Next question is human proteins can also made by using bacteria and that have been genetically modified. How a gene can be transferred from one organism by using genetic modification. So you know that in bacteria there is a circular DNA. This is called the plasmid. So we use the plasmid, and we also use some enzyme that is called restriction enzyme. These are like scissors, and they cut the DNA of the human and the gene that we desire to put in this plasmid. So by restriction using enzyme, we cut the part of plasmid and then we insert the desired gene here like this. Yeah, and the important thing is the sticky ends. So these here are the sticky ends, and which has the complementary base sequence. That is the important. And we have another enzyme, ligase. So ligase enzyme, when we put our gene sequence here, so the ligase enzyme will just stick it back. It is used to combine the genes and the plasmid of the recombinant plasmid. So it is put back into the bacteria, and then the bacteria can grow and make our desired product. The next question is, some people are worried that GM plant could pollinate wild plants. So how pollination between GM crops and wild plants can be prevented? So if we are prevented two different crops and we are worried about they could pollinate the other species. So the simple way is we can plant them far away, so their pollens and cannot mix, and because the distance is big, so the plant cell can separately and just far away from the wild field. State two advantages of the GM crops other than insect resistance. So the advantage is that they can give us high yield, more products, and they have resistance to the insects. So we use less use of pesticide and save money. Okay, so next question is that xerophytes. Xerophytes are those plants that actually grow on the soil. They have some adaptive features. So for the dry habitat, adaptive features mean they have something that allow them to survive on this dry habitat. So what is the term adaptive feature? Adaptive feature is actually inherited feature. It is transferred from one generation to the next. That actually help the organism to survive and reproduce in environment. So that is the definition, and these are the three key words that can explain the adaptive feature. Next picture, they show us a xerophyte plant and a cross section of madam grass leaf. So that you can say it is this kind of plant, they have a really adaptive feature to survive in this kind of environment. They ask you to write the visible features as a xerophyte plant. So if you can see their stomata, they are sunken inside. So stomata are very sunken inside, so they can reduce the rate of transpiration, because stomata, when they are open, they lose water, and in a dry condition, if the stomata are outside, they can lose more water, and this kind of activity can lead the plant to die and cannot survive. There are also, you can see some hairs like here. So this hair can actually trap some moistures in the atmosphere. So this can also help the plant to decrease the rate of transpiration. And they have in the upper surface, they have a very thick cuticle. It's a waxy layer that actually helps the plant to save water. For the 2 marks, you can at least write 2. Next, there's a The mean cell wall thickness of epidermal cell from one species of xerophyte is 1.276 micrometer. So convert 1.276 micrometer to millimeter. So you know that micrometer is equal to 10 power minus 6 and millimeter is 10 power minus 3. So when we convert micrometer to millimeter, so we add some more digits on this side, so it will be 0.001276 millimeter. Next question was, state the name of the carbohydrate molecule used to build the cell wall. So we know that the cell wall is made up of cellulose in the plant. It is strong and hard. So just one way of the stem of the xerophyte is adaptive for dry plant. Xerophyte, because they need more water, so they have a large and juicy because they need more water to stay alive in the dry environment. So that is the adaptation in the stem, because they ask you to what is the stem look like. The next, this shows the two species, species 1 and species 2, and asks us to write two root system they show here, and how each of one is an adaptation for living in dry environment. So if you see species 1, it have a lot of roots, but they are spread near the surface, upper surface. They can absorb more water, but only on the upper surface of the soil. But species 2, it has a long and deep root system that can absorb more water deep side, underground. So that is their adaptation. Question number 5, they show us a root hair cell, and they ask us to explain how nitrate ions are transported in the root hair cell. So if you see here, these are the nitrate ions. So when we see in the picture, the nitrate ions are higher inside the cell than in the soil, because you see more eggs are here, and because we need carrier proteins on the cell membrane to actively transport the nitrate ions against the concentration gradient. So we have learned there are three types of transport, like diffusion, osmosis, and active transport. So osmosis and diffusion is don't require energy and down the concentration gradient, but active transport is against the concentration gradient. So this kind of transportation will require energy, and energy is supplied by mitochondria, the powerhouse, and we also need the carrier proteins for the transportation. Next question is, two ways the nitrogen fixation occur. So nitrogen fixation, the one way is lightning during the rain, and the other way is nitrogen fixing bacteria. They can fix the nitrogen in the atmosphere. Next process, they want us to talk what is the process that moves water molecule into the root hair cell. Remember, the only difference between diffusion and osmosis is that osmosis is linked with water movement. The diffusion is also from high concentration to low concentration. Osmosis also from high to low concentration. But in osmosis, we consider the water molecule through a partial permeable membrane. That is osmosis. Next question, there's difference and similarities between structure of the plant root hair cell and bacterial cell. So the difference, if we talk, plant cell have nucleus while bacteria don't have any nucleus, but they have a circular DNA. We can call they have the plasmid. Plant cell have cell wall made up of cellulose, but bacterial cell wall is not made by cellulose. It is made by peptidoglycan. When we talk about the similarity, both have cytoplasm, both have mitochondria, both have the cell membrane, both have ribosome, both are the living organism. But we are talking structurally, so the next question, they show us the effect of pH on activity of two different protease enzyme. Remember, the effect of pH. At high pH, the enzyme become denatured. They only work optimum, best at optimum pH. There are different range of pH values, and after 60 minutes, the area of the jelly that has been broken down by L enzyme and enzyme M recording at each pH value tested. So if you see this graph, there are two type of enzyme. There is the enzyme L and enzyme M. So if you see this enzyme L, it first the activity is less when the pH is 1, but at pH 2 the activity is very high, is optimum. But when we increasing the pH 3 or 4 or 5, it becomes 0. Its active site activity denature and it's no longer fit its active site, and it cannot work. But for the enzyme M, at 5 you can see its activity is less, but when we keep increasing the pH, its activity increase. And you can see at pH 10 the activity is best, is very high. So the question asks you describe the result of this investigation. So the same thing, you can say optimum pH for L is 2, and it is lower for the M, and M functions at a wider range of pH from 3 to 12 than L, that is from 1 to 5, because enzyme likes the lower pH and M enzyme likes the higher pH. The area of jelly broken by M remains 0 millimeter square from pH 1 to 3, and then increase to 80 millimeter square from pH 3 to 7. The same thing I just explained in this graph. Area that is broken down by the L increases from 80 millimeter cube to 160 millimeter cube, that is the peak, the optimum, and from pH 1 to 2, and then decrease to 0 from pH 2 to 5, because every enzyme needs a specific pH to work. If that pH becomes too low or too high, the enzyme stops working. So the result of enzyme M at pH 2. So if you see this enzyme M at pH 2 here, so its activity is almost nil, because the low pH denature the enzyme. So the active site shape change. It's no longer make the complex. So it's not working. So if you suggest the name of the enzyme L and the location, because L like lower pH and must be present in the stomach, and in the stomach we have the enzyme pepsin.